So now I'd like to invite the panelists for the Biomechanisms panel up. They are Tim Bullock from the University of Virginia, Kathy Ferrara from UC Davis, Nathan McDonald from Brigham and Women's Hospital, Krit Moonen from University Medical Center Utrecht, and Xin Xu from University of Michigan. I just wanted to first ask each of you to share kind of your preferred mechanism that you're working most on, and how you really see the clinical utility for this specific mechanism moving forward. We can start down with Tim at the end.
Well, I only work with one mechanism at the moment. So it's the microbubble approach, using the cavitation effects of microbubbles to augment immune function in the tumor microenvironment. So there is a couple of very important elements associated with this approach that we think are going to have need to be explored tremendously before there's going to be direct clinical application. We still need to understand fundamentally what are the biological properties in the tumor microenvironment of causing cavitation based on, and the energy deposition that occurs with microbubble disruption in the tumor microenvironment. We're really at very nascent steps at this point. Taking that thought the whole way down the pipe to a clinical translation, it's my understanding that in the community that bubbles aren't nearly as progressed as far as the FDA is concerned as some other technologies, and so there's going to be some interesting barriers of entry at that level. But as we discuss further, I think there's going to be an awful lot of ancillary opportunities that are driven by the biology of bubbles, which may be parlayed into ultimately better therapeutic effects, but we can discuss that as we go along. So those are sort of the interesting elements, but also some of the barriers.
So I would say that our lab is fairly agnostic in terms of the ultrasonic mechanism, that depending on the question that we're trying to answer, we are interested in integrating various ultrasound physical mechanisms. Oops, maybe that wasn't the right answer. I guess the other thing I would say that maybe is driving us at the moment is figuring out how to combine ultrasound mechanisms in a reasonable manner with new biochemical and other approaches to treat disease. And so I think one of our themes is to learn how to optimally combine with things like checkpoint inhibitors in immunotherapy or chemotherapeutics of various kinds.
I mean, we work on a variety of mechanisms, but I think by far the most that we work on is using microbubbles and ultrasound to disrupt vascular barriers, primarily in the brain, for the delivery of drugs. I think it's a really sort of a unique technology that we're not trying to replace something else with focused ultrasound. I think this is something pretty unique that we can do, and there's a huge need for this, and I think that we really could open up the central nervous system, tumors and other diseases for treatments that all of these great nanoparticles and targeted drugs. If we want to use them in the brain, we have to deal with the blood-brain barrier. So I think for me, that's the one that has been the focus of my work.
Well, for me, I think it's clear that also from this conference, there is not a single mechanism. There are so many really promising approaches. But still, for me, if there is one to favor, I think as of this moment, on the short term, it's still ablation. I think the impact there, the clinical impact, can be reached fairly quickly, and I think that's, for me, if I would favor one at this point, it's still ablation.
For me, at University of Michigan, we have been working on histotripsy. So yesterday, Kathy and I were talking, and she said, "You'd better say histotripsy, otherwise you will be kicked out of University of Michigan." So that's certainly my favorite mechanism now. Histotripsy uses cavitation to fractionate cellular structures and tissue structures, and it can be used for a number of things. And we have been investigating histotripsy mainly for cancer and the cardiovascular applications. I think that right now, because it's a relatively new technology, in terms of technology side, we have been making advances, but because it's so different in terms of mechanism from the established ablation technology, mainly based on thermal effect. There's a lot of unknowns, uncertainties, and we don't have all this established human data. What will happen to human in those different diseases? What are the associated complications, and so on? So I think those are the things that will be critical questions, and that need to be answered before we can reach the clinical translation later.
So many of you, talked about the fact that you're using several mechanisms of focused ultrasound depending on the clinical case. So I wonder if you want to, share your thoughts on focused ultrasound as a treatment platform that by providing these different mechanisms enables the treatment of different diseases, and sort of how you envision using focused ultrasound as more of a platform in clinical use in the future. Does anyone have thoughts on that?
Oh, I think it's sort of an ideal with having all of these different mechanisms really opens up the idea that we could do all sorts of therapies in one patient. So we could ablate a tumor, we could go around the margins and enhance drug delivery, or radiosensitize the tissue, or both. So I think as a platform, if we have a variety of parameters that the devices, the commercial devices, can apply safely, I think that it can be not just one. I think in one patient can have multiple treatments with focused ultrasound.
Yeah, I agree with Nathan. I think I don't think particularly one particular mechanism within focused ultrasound would be the one magic cure for everything. So I think most likely a combination of different mechanisms, like Nathan was talking about, would be needed depending on what disease stage you're treating. So maybe ablation may be used for treatment for solid tumor has not spread yet, but most likely if you have infiltration and metastasis and you want to combine with drug delivery and even immunotherapy, I would even say that I think we should keep our eyes open to potentially combine focused ultrasound approach with other approaches, radiation therapy and other therapy approaches, with just a common goal that eventually we want to just treat cancer, and what would work the best, and that continue collaborations and exploring together, I think would be the key.
So sticking with that, on that line of focused ultrasound combined with other approaches or other therapies, obviously for the treatment of cancer, we see that. Do you think that in those cases, focused ultrasound is potentially a kind of first line of defense, or as a primary treatment that then could be other things coming in to sort of supplement that, or do you see it more as an adjuvant therapy? Maybe, and that's could be different depending on the different disease, but maybe talk more about focused ultrasound being used in this combination approach, where you see it fitting in, and then also where we're at now with studies to determine where it fits in, and what kind of research we still need to do in that area.
I think we are so just beginning to scratch the surface of the local versus systemic effects of the various different ultrasound platforms that it makes it difficult to predict at this current time what stage ultrasound will be a standalone monotherapy. I suspect, as we're learning in just about all other forms of immunotherapy for cancer, combination therapy is going to be the way forward. So there's going to be a patient-driven selection of the type or the disease state in the patient that is going to define the type of ultrasound that ultimately is going to be most beneficial for any patient. So if the patient, for example, is got a systemic disease, that metastatic state is going to require a very different therapeutic approach than someone who's got a well-defined primary lesion that could be tackled with an ablation approach. And some of the work that Nathan and Kathy are doing of understanding how modulation of tumor vasculature, allowing it to be more permissive, that it defines that you're going to be using a combination approach with a chemotherapeutic or an immunotherapeutic. We don't really understand any of the parameters by which antibodies really are decorating the immune cells in the tumor microenvironment. So, boy, there's a lot to explore before we can answer that question.
But I think it depends a little bit on the application. I think the strength of that is of these combination approaches, I think from this conference are in the long term are really clear. But that's long term, and I think there is also, I mean, to the outside world, for here, I think we are all fascinated by these new developments, and I think we expect a lot from it. But on the short term, I think there is still a bit of a risk. I think that it's a confusing story to the outside world. And if you look at some of the direct effects, just to take one example, is by different groups on osteoid osteomas, the progress has been very direct, and it's a single treatment with only MRI-FU, and the results are pretty spectacular and convincing. I think for us as a field to be accepted by the rest of the community, I think we also need some of these direct, simple successes like the one in osteoid osteoma, just to name one.
Yeah, and speaking of ablation, we're aiming to replace surgery most of the time, and at least in most cancer, surgery alone is very rarely the monotherapy. So I think for ablation at least, we're always going to have probably, or at least most of the time, a combination therapy. And I think one of the things that we haven't explored as much as we could would be the interactions between ablation via different mechanisms and radiation. I mean, I think that there are synergistic effects, that we know from hyperthermia that could apply with ablation, but there's less work out there to really understand how these two things interact. And it really could be beneficial for us.
I would just reiterate what I think people are saying, that I agree that in most cases, ultrasound would be part of a combination, but to move it towards a first-line combination in timing is something that's really important and that we should be shooting for.
Okay. Thinking then about challenges as we're talking about new mechanisms, I feel, well, first maybe let's think about ablation with focused ultrasound. Where do we think we're at with ablation now? We're doing a lot of different clinical trials looking into new indications. Is there still work that needs to be done in understanding more about the safety of that kind of mechanism, or are we beyond all of that, and now when we're thinking about safety of mechanisms, we're just moving into these other areas?
Well, at least I think in the brain, I think that we still have a lot to learn about that. And again, going back, if we ended up doing this, I think we need to be more careful. I think we understand well, thermal mechanisms and how it relates to temperature. And we all know about technological advances. We need to improve that with thermometry and better systems and that kind of thing. But I think that we need to be careful in the brain about potential interactions between other things that we don't see in our animal studies. What is the patient history? Because we kind of push things sometimes, especially now that we're in the clinic, and there can be really challenging patients, and you want to get that lesion in the place, and you push it up and up and up, and you start to see the dose accumulate. And so far, we haven't seen the adverse events, but I think they'll come. And part of that might be from factors in these patients that we're not anticipating. So I think there is still some to learn even with thermal ablation.
I think it depends a lot on the field. I think that what you see in the prostate, for example, it's already a very advanced technology for the abdominal work. For ablation, it's still, I think, with respect to technology, still quite a bit needs to be done, I think, on the side of hardware and so on. But with respect to the mechanism of ablation, I think the biological mechanism, I think that's what already quite advanced, but technology-wise in some areas still need quite a bit of an area that needs attention. Another thing, for example, in breast ablation too. we have seen the field of fibroadenomas progressing quite a bit, but not the breast tumor, the malignant tumor field. So there's still quite a bit to be done.
I think the compared to thermal ablation methods as relatively established, especially you can draw from RF ablation, microablation, this other thermal-based ablation with lots of the human data, new technology that's cavitation-based, such as histotripsy for ablation, is still quite new. The biological effects are not quite understood, and there's not much, not much human data out there to study, especially in specific disease states. We started to look into in the animal states where, for example, we fractionate tissue, right? So you have all this acellular debris left in the organ. So, in abdominal organs we study, they seem to be reabsorbed by the body over time. But what would happen if you actually leave that acellular debris in the brain? We don't know. In other parts of the body, what we have seen is that we don't create much of bleeding. So it seems like the cavitation is also releasing coagulation factors that it actually coagulates the blood vessels so that you don't get much bleeding. In the brain, the latest study, we also seeing that we haven't got no bleeding outside the lesion. There's some inside the lesion, but is that already sufficient to cause a major complication in the brain? We don't know either. The good thing is, the acellular debris, they are liquefied, so they're like liquid. So in the brain, we have tried that we can actually just use catheter to aspirate them out. So there are possibilities. But just a lot of things that need to be investigated. But the reason we are looking at the new modality like histotripsy, because there are advantages such as, take brain, for example, we can actually, because we're not doing heating, right? We're actually using very low duty cycles, so the heating is dramatically reduced. So we are able to treat within 5 millimeter from the skull. So that will allow us to treat majority of the cortex tissue to cover where the brain tumor would be. So there are certainly opportunities, but there are a lot of unknowns that need to be addressed, at least for cavitation based, such as histotripsy.
For ablation, I think still the interaction with the immune system, and particularly the effects of the pro-inflammatory effects that occur around the rim of an ablated site are still things that we need to better understand and understand the implications of.
Okay. As we kind of move forward with any of these mechanisms of focused ultrasound and exploiting them for clinical utility, what are some challenges that you see in really bringing these to patient therapies? One thing I'm thinking of, and not just like regulatory challenges, but I know in the case of using focused ultrasound to open the blood-brain barrier, there is this sort of prevailing thought in the community that opening the blood-brain barrier is not a good idea. What are some things that maybe we as a research community can do to try to combat some of those prevailing theories? Is it more collaboration with the neuroscience community, or more education on this? Do we need continue to get more safety data? Maybe Nathan would be a good person to first address this.
I mean, I think there is a history of blood-brain barrier disruption for brain tumor therapy using mannitol. So I think for brain tumors, we can point to that and the successes that they've had, and hopefully overcome, at least for that application. I mean, when we start to talk about other applications beyond brain tumors, it is going to be an issue, and I think there's always going to be skepticism until we do the clinical trial. I mean, because we've done, repeated treatments in all sorts of animal models and disease models, and we don't see at least significant side effects. So I think only time and experience is going to help with that. Maybe we could reach out to some of the more skeptical groups and talk to them and ask them what would, convince them, what kind of data do they want to see. Maybe we already have it, maybe because they don't know about it, but maybe that would be an approach.
I think there's always the inevitable risk-reward status. So if you, in the case of GBM, you have a defined timeline for your patient, and the patient and their physician will have to have a very candid discussion on any well-controlled phase 1 clinical trial where you're assessing safety as your primary endpoint. Is this a modality that the patient would like to include in their treatment regimen? It's going to be a very different discussion for ablation in a GBM or a melanoma brain metastasis compared to a stage 1 breast cancer patient, where they have a lot of choices. Surgery is often curative in that case. So it's the context. So as we move to the more complicated scenarios, it's going to be driven by the patient. And so we have to reach out to the practicing community and educate the patients, but also design. This is where the foundation can come in, I think, be fundamentally involved in the design of very, very good phase 1 clinical trials. And nothing will happen until we get those trials off the ground. We can model anything in mice, but it's so different when we go to the patients that's the fundamental first step.
Yeah, I still see also the hurdles because of it's a paradigm changer. The clinical field is not familiar with the kind of technology, so how to address that is, I think, to really reach out and be present on different conferences and education and so on. I think there's also maybe a case to be made to link up with radiotherapy, because the approaches that we are doing, they resemble quite a bit that field, and I think that also there's a really good effort ongoing from the side of AAPM. To a task group that actually writing a white paper on the HIFU technology, how to do that, how to address safety, how to address quality assurance, all of that. I think all of the different parts have their importance, but it's still very important, I think, to reach out to these communities.
Kind of following on that, and one of our themes as we go through the day is also talking about collaboration. And I'm wondering your thoughts on and your experiences with collaboration, particularly, not just within the focused ultrasound community, that's incredibly important, but also in communities outside. Obviously, Tim is an immunologist and has now, I think, been very involved with the focused ultrasound community, but I know all of you have collaborations like that where you're connected with different clinicians or biologists, and I think for the community and particularly for the younger researchers, talk about the importance of that when you're really trying to explore something that's outside your field of expertise.
I think you put the burden on me there, Jessica. I wouldn't be here today if it wasn't collaboration, right? So I have my niche. I could study the elements of the tumor microenvironment, the basis by which T cells become dysfunctional, which is really the elemental, the essence of our laboratory. But it's the opportunity that you get by having conversations with people who don't work in your field, where we make these serendipitous discoveries that generate new fields. So it wasn't that long ago that tumor immunotherapy was a failed art, that it wasn't going to be successful, but persistence in understanding why failures happened allowed us to understand that these notions of adaptive resistance in the tumor microenvironment. So now we have opportunities for patients. And I look at this evolving environment here with focused ultrasound in exactly the same manner. We know that X number, 20% of patients really receive benefit from checkpoint inhibition. How do we explode that number to everybody? And I wouldn't be here talking today if I hadn't heard Rich Price giving a seminar at UVA, talking about opening using microbubbles to open environments to allow drugs to more penetrate. And that's just one of the hypotheses we have about T cell trafficking to the tumors. So it allows opportunity. You raised one very important point about these vertically integrated units within research. So at UVA, our structure is really, in the cancer center, is really becoming basic science, translational science, clinical science? And can we integrate all of those to make very productive elements? And then how do we bring in physics? How do we bring in the opportunities that come in from elements that us biologists aren't always thinking about all the time? So I think collaboration is so important, and you probably have similar experiences at your institutions.
Yeah, I would say that collaboration it certainly is a key. I totally agree, and I would think trying to speak from clinical application point of view. We're all working on clinical application with eventual goal to actually do clinical translation. So collaborating with a clinician has been incredibly important from right beginning. To hear from that clinical perspective, what are the critical things, critical issues, when we're trying to treat a disease, we go in, we're trying to treat a liver tumor, but what are the clinical complications and what are the existing field, right? Where can we make the biggest impact? How can you design your study to address the issues that clinicians are really concerned about? And really an issue in the current treatment regime. And those are things that if we only work as engineers and scientists and basic scientists in the laboratory, we wouldn't really know. So it's almost like if we don't think as academic. We're researchers. We don't really think from this point of view. But if you are in a company designing a device, developing new technology, what do? You trying to think about users, right? The clinicians are the users. So you actually design based on the user requirement. So user come in to tell you, what do we really need? And I think, for clinical application point of view, the clinicians, they really bring that perspective. Then we talk about the combination of mechanism. That's where really we need all the expertise to contribute to that, and beyond our own expertise.
I couldn't agree more that the collaborations are really crucial. We're trying to set up programs in drug delivery and in immune system stimulation, and without really directly collaborating with experts like Tim, all of people in our field in Utrecht at our place. But it would also be good, I think, to that there are funding mechanisms that they take that into account, that these collaborations are so crucial, and that there are opportunities to really set up combined programs with the different groups with their expertise.
One thing that we've experienced, or at least I personally experienced, is we do all of these different new effects, and they seem to be really fascinating, but it's been hard to get biologists and other related scientists to come and work with us to try to understand what's happening. I mean, we're a small field. We don't publish our papers in Science and Nature, and a lot of times the biologists are used to that more than we would be. So I think finding ways to better engage with that type. And I've been happy with this meeting, seeing more of the sort of deeper investigation of these mechanisms that we're exploring now, not beyond just the cooking tissue.
Yeah, I just to echo that collaborations are really important, and I think many of us also feel that we have so many people at our institution who would like to try something with ultrasound that it becomes challenging just to keep up and to decide what are the most promising directions or questions to ask. It can be really difficult. I also wanted to say that I think the foundation is playing a really important role in bringing people together in a meeting like this, but also in starting to talk about things like shared models and being able to replicate experiments and findings across different institutions. I think right where we are right now with some of these really complicated questions in immunotherapy or wherever, that this is going to be tremendously important, and that the role that you have to play is really super important.
Yeah, I can't overemphasize that lateral collaboration element. I know this is the Focused Ultrasound Foundation, but my sense coming from the outside is there's very fragmented approaches, and there's a lot of, you could consider them competing technologies or competing approaches, and to prevent siloing of development, it's going to be so important to have these cross-approach understanding of inflammation, of cytokine release with each of these individual modalities, and that's only going to come by lateral collaboration, sort of setting up study groups. I think that'd be fantastic.
I also actually just want to follow up on one more point is our collaborators there in different fields, they take our study then to talk to their community, and that's also the way to spread our impact beyond just a focus just on the community. And I echo on what the other panelists talk about, that at this symposium we see a lot more people just beyond our small own community, and we, kind of the previous question in terms of how can we reach kind of the clinical widespread use eventually, that we need to do the education, right? We need to do education so people know about more. If you have clinician and you have our spokespeople in that field, right? And it's the more influential figures in that field, then they are more willing to listen. And that's, one way we can reach out through our collaborators to talking to their community.
Great. I wanted to open it up to the audience and see if you have any questions for our panel, or we can keep talking. All right. So thinking about challenges, moving your research ahead, what are you seeing now as the biggest challenges as you're still exploring the different mechanisms that you are, but also clinical translation? Maybe each of you can give your thoughts on that.
I guess the time that it takes to do many of these studies is, especially as we get towards, the really encouraging thing is that we're really at the point of looking at long-term survival and long-term effects in many cases, and so the timing becomes really important. And also the complexity of some of the biological assays, and so this is another area where it'd be great to share more information, but we're beginning to look at more and more complex questions and systems and assays, and so sharing becomes more important.
I still think one of the most challenging things is the clinical translation on these different topics, both with respect to new hardware, or if you work on drug delivery, the nanocarriers, how to really get them into the clinic. That's a big hurdle. So with respect to especially the regulatory aspects. So these are, I think, one of the biggest ones.
Yeah, I think we're moving systemically towards patient treatment, and the frustrating thing has just been how slow it's been. I think we're getting there now. We're doing patients now. We're meeting with FDA, and just to get something out there going so that people see what we're doing, I think that's going to be important. But I mean, finding out how to pay for the different trials, we could come up with lots of trials to help people, but getting funding for them is an issue always, and the regulatory is a big unknown also with these combination products.
My sense is a little bit more granular than these higher level problems, particularly when we think about where we are advocating the use of focused ultrasound, particularly for brain metastases or primary brain disease. We know so little about brain immunology. We know so little about how the types of inflammation, what sort of mechanisms of immunosuppression that are going to occur, what are going to be the systemic effects of releasing cytokines in the brain, or even nervous stimulation in the brain, what's that going to do at the systemic level. So it's a little bit more granular, and I anticipate that we're going to have to spend quite a lot of time really dissecting what are the impacts of either thermal or acoustic energy in the brain.
Yeah, so we have for the histotripsy, there's the first clinical trial in prostate. We collected some human data, and we are trying to pushing a device through clinical translation for the cancer treatment. For the device, I think it just really takes a lot of resources. Not only is the regulatory part, which is really a big hurdle, and the clinical trial, and even just the device itself. So really the resources, and also because it's such a new technology, we don't have that many chances to just try out. Right? When you get there, we need to get the first trial and really trying to make it as successful as possible. So the first step to make it a success, big success, I think is critical to the future and expanding it beyond that. And in order to do that, it just means that we have to be really careful about a lot of things. And again, it just takes a lot of resources and time. And it's I found it interesting that the investors who work in the medical device field, you think they should know by now, by doing so many years, it just takes a long time for the building medical device and getting into clinical trials, get the right data. But everybody has a high expectation for return on the investment, right, on the short term, and trying to push things down, in my opinion, sometimes not giving the highest chance for success. But I don't know if this is something that our community as researchers can address, because it really, the clinical translation, we're just kind of the starting point to really move beyond that. And we need everybody at the table really trying to, I think that's clinical translation, in my opinion, is still the hardest thing to do.
Mm-hmm.
Yeah.
I think beyond the commercial and us, I think that finding the MDs who are willing to really dedicate themselves to this. I mean, we don't all have a Peiji or a Wadi or an Alexandro at our site. Right. And I think trying to identify those people and bring them, grab them for focus ultrasound would do a lot too.
The champions,
Right.
yeah.
So a mechanism
yeah.
to clone these three.
Oh, well, that's a good idea. That's a good idea. So from the panel perspective, are there, do you have topics that you'd like to bring up now, share with the community, or any other comments that you wanted to make?
I think overall, I think I very much enjoyed this meeting, and I think I echo the previous comments. I think the Focused Ultrasound Foundation, I think, did a terrific job, and also the journal, I think, to have a combined journal, because we are a little bit spread out in different organizations. With the EAST2, the Focused Ultrasound Foundation, the European First Foundation, also things that we have now a combined journal. What we heard this morning, that also the European First Foundation would like to see this in one journal. I think all of these things help, I think, to consolidate the field and to work together, that also to the outside world, that we have that we speak with one voice. I think that we will have a better impact.
I actually think you make a great point, and this could be a really great utility of the journal. We are so dependent on publishing successful results from NIH funding, that there's an opportunity here as we talk about the different platforms and the different barriers of entrance and the different success rates potentially, that we encourage the community to publish even, not anecdotal, but even descriptive results in the journal, so that we build out that platform of base knowledge which is easily shared, that when things don't work as expected, that's got to be published just as much as the great success stories. So I think there's an opportunity to be very transparent here, very early on, and I think a lot of translational and deployment of technologies and approaches have been delayed throughout history, simply because people are not transparent with each other at a very early point, and so you don't get the synergies that you would like to get from a community of investigators that are learning from each other all the time. And if you keep the ultimate goal, which is treating a patient, as the endpoint, and some of the barriers we talked about, how to get something to the patient, the clinicians, getting the patients involved in the selection of therapies, then if you take that long-term view, then there's an opportunity to work together as a community. Sure, we learn a lot from our own investigations and our own fascinations with the biology and the physics that are going on here. But if we keep it transparent, we keep publishing stuff that is not necessarily successful, as well as the success stories, we'll make much more rapid progress. And that will be exciting for everybody.
Yes, one other thought. This was touched on earlier, perhaps, but being able to have sessions at other conferences and is something that we really need to think about. I know some of us have been really working on keeping visibility within, say, the World Molecular Imaging Society, but I think other avenues, especially more clinical avenues, one of my favorites is American Association for Cancer Research to, to really bring up the visibility both of imaging but also of interventional therapies within that community is something that would be a great thing to work on, for those who are particularly interested in some of those areas. But I think bringing this kind of visibility or at least some visibility in some of those other venues is something that we should also be really focused on in the next couple of years.
Yeah, and just sort of a plug for what we're doing at the foundation. So we're doing a lot to try to connect with other organizations. We have, in cancer, we have a great connection with the Cancer Research Institute, which is devoted to cancer immunology. And we were thinking specifically about AACR and perhaps using CRI to, connect with AACR, but things like that we're definitely are on our radar to try to do to get more visibility for this community in specific areas. And following up what Tim was saying about publishing negative results, we've heard from a couple of people at this meeting that perhaps the foundation could help be a repository for some negative data even in certain areas like in the immunology experiments that are going on or in BBB or things like that. So we'll think about that as well. And again, we always want to hear from the community as to what we can be doing better or what we can be doing to help facilitate the work that you're doing. And so again, if anyone in the audience has any questions, now's the time.
So I was wondering what the panel thinks about whether it's a big problem for the field when you're investigating quite complex bioeffects, that people are using different devices and different settings, different microbubbles, and is there a better way that we could report or standardize these settings so that the bioeffects, there's less noise when we're already investigating something that's quite noisy and quite complex?
Yeah, sure. I mean, this is clearly a very good point. That is, like what is it called, Gail Terhaar. Gail is very much advocating of the standard, developing proper standards. That is the way that you do the parameters and so on. It's very deep in many different fields. I think, Nathan's field and the blood-brain barrier is clear, but also overall zone operation. It's so much dependent on the parameters that you are using. So a really good way standard way of reporting all the different parameters is, I think, a really key issue, I think, in this field.
Yeah, so I think just follow up as a quick point that Gail, the standard that established, if everybody can report all the acoustic parameters following list with the pressure, the intensity, the pulse duration, frequency, duty cycle. So everybody, when they look at the paper and the microbubble you mentioned, the brand and size, and if those detailed information can be reported, then when you see it, even though there will be some variation and differences, but at least you know what could cause that. So I think reporting and the following kind of standards report all the acoustic parameters and all the other parameters would be important to at least that you can analyze the difference across different publications.
And I think that's particularly important too, as people outside the traditional focused ultrasound field are entering into this type of research that don't have the acoustics background and don't have the physics background. So it's important that as a community, we not only develop these standards so that we understand them, but that they're also understandable to biologists and others who are trying to get into this research.
I think now that we're moving a lot into these bubble-based therapies, we need to come up with new standards for reporting, cavitation monitoring and cavitation dose. I don't think that we need to characterize our receivers and know their sensitivity in the focal area. Otherwise, I think it's really hard to compare our results to other people's results. Right now, we're just sort of having a binary, do you have this type of cavitation or not? But I think going forward, we're going to want to have cavitation at some predefined level and I think that's something we could work on.
Any other questions? Oh.
Hi, hello. Maybe this is coming more for the foundation, or also as some community. Some of us, for example, we are really true believers in collaborations, and we help each other. For example, we develop some software libraries of tools or procedures, and we exchange between each other in order to try to harmonize, we standardize, or avoid that other teams repeat from zero, for example, building an MR thermometry library from scratch. So we make a lot of efforts in order to support, but there's no like an overall structure that could probably might benefit all the community. Is there a potential opportunity, maybe guided by the foundation, to fund and support this type of developments to create like harmonized tools that everyone can benefit?
Yeah, I think that sounds like a potential opportunity. We are always looking for, different platforms or things that people are developing that could be useful for the field as a whole. So if you specifically want to come talk to us, I'd encourage you to, and we can see if there's something there that would be good.
Yeah, I have a precedent. For example, we developed a couple tools to control, for example, Philips scanner and the Sonalepe therapeutic table, and we share those tools with many centers. We have great, fantastic collaborations. Things we share those tools freely, no question asked. So, but now we are thinking of how to take it to a bigger level, to extend the tools that we can really all together enhance. Thank you.
Thanks.
You got time for one more?
Yep.
Yeah, so this is Matt Myers from the FDA, and I wanted to second the vote for a repository of studies that don't work. From an FDA perspective, what we want to know is not just what works, but what doesn't work, because when the submissions come to us, we need a balanced picture. We want to know how far you are from the conditions that didn't work. We see a lot of submissions where people say, I've got this good idea, it's worked in this study, approve me, please. And we can't do that. We have to have a broader perspective on just the whole state of the art for these different procedures. And I think everybody's acknowledged during this symposium how complicated these mechanisms are. And so to the extent that everybody here can help us put their particular study in perspective of what works and what doesn't work, and what we know and what we don't know, including this earlier question about standardizing your results relative to the parameters used by other studies. I think that would be very helpful and help us to expedite review of all these great ideas that we've seen. Thanks.
Great. Do we have any other Quick question. Yep.
So we have a lot of puzzles in biology when it comes to us applying ultrasound to a biological system, right? So if you had a magic ball and could uncover one puzzle today, like what would it be? This is a question for each one of you, and why? There's many.
I think for me, for my perspective, my parochial view of these things, it would be the inflammation. So the extent to which ablation versus histotripsy versus microbubble cavitation, what are their discrete impacts on inflammation in different environments? So a skin tumor versus a brain tumor. So what are those parameters, and what will we learn from those in terms of the capacity of the cells that I'm most interested in, T cells, trafficking into a tumor? If we can get a very good map of that, then we'll have an opportunity to make rapid progression in many elements of immunotherapy, whether it's adoptive transfer therapy or checkpoint inhibition or regulatory cell depletion. That's very parochial.
Well, yeah, and I echo some of the same questions. We're really interested in whether and why our T cells are activated or not, and under certain conditions, and how to control that.
I would like to know if local therapy is when or if it's ever a good idea, and what so we can direct our efforts to those things.
Nothing to add specifically.
Since it covered the immune response, which is, what I would love to know, whether histotripsy actually induces immune response. I would add to see that I want to also know that what happened to, I talk about coagulation hemolysis effect, what does histotripsy do, and hemorrhage, so kind of related to blood, when we treat tissue.
Thank you.
All right, I think that is our time. So I want to have all of you thank our panelists.